Today it is my pleasure to introduce Nick Banovich, who is coming here all the way from across town, downtown Phoenix, from the Translational Genetics Institute. Translational Genomics Institute. They don't just do genetics, it's genomics now. So Nick actually did his undergraduate here at ASU. He was in the School for Human Evolution and Social Change working with Anne Stone. He went off and did his PhD at the University of Chicago with Yoav Gilad in human genetics, and I'm afraid I don't know where you did your postdoc. Okay, and then he went and did his postdoc at TGen, where he is now a PI running a fantastic lab using a lot of cutting-edge genomic tools, both single cell and bulk, trying to understand human variation, genetic variation and how that impacts disease susceptibility across different tissues. So with that, I'll let Nick take over and give his talk.
All right. Well, thanks, Noah, for inviting me out to talk. And yeah, like Noah said, I did my undergrad here at ASU and actually took my first genetics course in LSE 104 across the hall. And so I was a little worried I was going to be in there and have a little PTSD, but yeah, it's always good to be here. And like Noah said, I'm at TGen, which is in downtown Phoenix, and we're a standalone nonprofit research institute. Do a lot of sort of genomics and genetics research, a big focus in cancer, although I'm not going to talk to you at all about cancer today. So I finished this talk yesterday, and I kind of compiled a bunch of new things that my lab's been doing. So this is the first time I'm giving this kind of iteration of the talk, so I don't know. We'll see how it goes. Hopefully it'll be okay. As you could tell from my talk title, I'm going to discuss some of our work in the lab on pulmonary fibrosis. And like Noah said, we use a lot of sort of genetic and genomic approaches to study a sort of handful of diseases in my lab, but we have a big focus on lung development and lung disease. And so today all the work I'm going to share is kind of an overview of the work that we have been doing and are doing and are kind of continuing to do on the lung. So if, like me, you are sort of unfamiliar with the lung, like I was when I started this work, I thought it would be helpful to orient everybody to some sort of just lung basics. So particularly around the sort of epithelial cells of the lung. So as you can see from this picture, the sort of lung is broken down into these separate lobes, and there's a trachea, and then these bronchioles and bronchi. And each section of the lung epithelium kind of has this different component of cells, with the bronchi, or the airways, being made up of these sort of basal and secretory cells. And we typically think about this as sort of the airway or the proximal epithelium in the lung. And then as you get out further into these branches, you have the alveolar structures, and these are the structures that are responsible for taking the oxygen that you breathe in and putting it into your blood. And this is really made up of two major cell types, the type 1 pneumocytes, or as we call them, AT1 cells, and the type 2 pneumocytes, or AT2 cells. And so the AT1 cells are really these thin, flat cells that are responsible for taking the oxygen out of the air and moving it into your blood. And the AT2 cells sort of serve as a stem population that regenerate the alveolar structures as they may get damaged over time. So we're really interested in this disease called pulmonary fibrosis, and this is a semi-rare, but still fairly prevalent disease that affects roughly 50,000 new people every year. It mostly affects older individuals greater than 55 or 65 years of age. And basically what happens is this alveolar space starts to get overtaken by scar tissue. And it kind of has this twofold effect on the lung. So as you might imagine, as you take a breath, right, your lung needs to expand and inflate to suck air in from the outside. And so as pulmonary fibrosis takes over, your lung loses that elasticity. It can't expand and contract to the same degree that it can normally. What also happens is as these alveolar junctions start to get coated in fibrotic tissue, you actually lose the ability to sort of move gas out of the air and into your bloodstream. So you kind of have this twofold issue where you're both not getting as deep of a breath for every breath, and you're converting less oxygen into your bloodstream with every breath. And the individuals who I've talked to who have pulmonary fibrosis describe it at the end of life as like trying to breathe through a coffee straw, that you just feel like you can't breathe, right? And so the median survival for this disease after diagnosis is 3 to 5 years of age. And the only durable treatment solution right now is a lung transplant. So there are 2 FDA-approved drugs. Both of them are targeted at the sort of fibrotic process, but neither of them actually change overall outcomes. They just slow disease progression a little bit. And they tend to have like fairly serious side effects, and many individuals can't tolerate them and end up going off. So just to elaborate a little bit on this idea of like what's happening to the alveoli, so you kind of get this really dramatic remodeling of the lung. So not only are you sort of getting scar tissue building up around these alveolar structures, but the whole kind of composition of the lung is changing, and some of these alveolar structures are getting completely destroyed and abolished. Some of them are getting elongated and get these really weird structures, and you kind of lose the normal composition and structure of the lung. And so it's a really, like, in some ways, unique disease in terms of how much remodeling actually happens. So just to orient you to what this looks like on some real tissue, these are H&E stains, and even though we have somebody from the Department of Pathology here, I understand most of you are probably not pathologists and probably don't look at these types of pictures, but this is a thin tissue slice from a piece of lung. And over here on the left-hand side is a picture of what a normal lung looks like. So you can see most of it is open air, right? Most of it's space. And each one of these little circles are these little alveolar structures where the air is coming in. And then some of those bigger structures are those sort of larger airways. And what you can see here on the right is what a lung looks like after at sort of the end stage of disease. Most of that normal alveolar structure is gone. Maybe there's a little bit of it left over kind of in the bottom mid-right of this picture, but it looks a little bit wonky. It doesn't have the same sort of structure. Everything's thickened. And for the most part, that the alveolar structures are just completely abolished. And like I said in the beginning, the current FDA-approved drugs are really focused on this sort of fibrotic process. And we believe, and I think others are continuing to, sort of our knowledge of this is continuing to expand and evolve, but a lot of people are continuing to think that this therapeutic modality probably has a low ceiling, that you're targeting the really last stage of the disease process, which means that you're not going to really affect kind of overall comprehensive change in the disease, and you're really just sort of targeting the very, very, very last step. So we think a lot of this dysregulation occurs much earlier and probably is involved with epithelial and immune cell dysregulation, and that to understand how to develop better treatments, we really need to focus on these earlier stages of the disease, not on trying to treat this sort of very end stage fibrotic process. And so we became really interested in looking at this tissue through the lens of this, at the time, which was 6-ish years ago, novel technology of single-cell genomics, right? And so if you are not sort of familiar with the idea of single-cell genomics, rather than taking a tissue and grinding it up in bulk and extracting all of the RNA, which gives you some composite average of all of the expression of all of the cells that you're looking at, you actually extract RNA from individual cells sort of in isolate across thousands of cells in parallel. And the most common way people do this and the way that we've approached this is using 10x Genomics, which is a company that has done this in a way that's very simple and works really robustly. And so I'm going to tell you a little bit about the work that we've been doing in the lab, starting with some of our single-cell genomics approaches and then moving into some of the newer work that we're doing in spatial transcriptomics. And my talk is going to really be in 3 pieces. So the first, I'm going to talk about some of our cell atlasing efforts, which was really just sort of profiling these tissues with this novel technology. This work is like pretty much complete. We've published a couple things, so I'm not going to spend a ton of time on that, but I think it's helpful to orient for the other pieces of the talk. Next, I'll talk about some of our work looking at genetic control of gene regulation. And this is work that's unpublished, but kind of close to a final story. And then last, I'll talk about some of the spatial transcriptomics work, which is literally data that I think some portion of what I put in here like came off of the instrument like 4 days ago. So very incomplete, but I think kind of fun and cool. And we've got Doug in the room, so we can see what he's got to say about it. Okay, so the first part of this project is cell atlasing, and this is work that was really driven by 3 people in the lab: Lynn Bowie, Austin Gutierrez, and then Lance Peter, who's my lab manager. And the idea was to just get a bunch of these tissues, use these single-cell genomics technologies, and understand what is the cellular composition of these lungs as they're undergoing this fibrotic process, what sort of dysregulation is happening within these cells, and then how does this compare to normal tissues. And on top of the work that we've done sort of for cell atlasing and disease, we've also contributed to some of these larger cell atlasing efforts that are just trying to categorize all healthy human cells, mainly this sort of human cell atlas effort. So this first study, like I said, is already published. It came out a couple years ago, but really, with a fairly small number of individuals, we only had 30 samples in this first study. We found some really interesting things. So the first thing that we found that was just nice to see is this idea of the proximalization of the distal lung. And so what we found is that as we looked at these sort of distal lung tissues, rather than them being composed as individuals who had pulmonary fibrosis, rather than them being composed of these type 1 and type 2 cells predominantly like they should have been, we saw this huge influx of airway epithelial cells or these proximal epithelial cells into these sort of regions that should have been alveolar. And part of the idea here is that the lung can't build an alveolus without first building an airway to it. So we think this is some aberrant repair mechanisms that the lung's trying to kind of build airways to then stack alveoli on top of, but instead of sort of correctly building the structures, everything just goes haywire, and you get this really large influx of these airway epithelial cells. But what was really interesting is we identified 3 cell populations that hadn't been previously described in the literature. And so we were really interested in kind of digging down on these sort of 3 cell populations, these cells that we called transitional AT2 cells, this other cell that we called KRT5 negative, KRT17 positive. You can tell I'm not very good at naming things. There's probably way better ways to name that cell type. And then another cell, there was the secretory cell in the lung, but a normal secretory, these normal secretoglobin cells in the lung tend to be double positive for both of these markers, and this cell was really predominantly positive only for these SCG, for this single gene SCGB3A2. So first, when you identify things computationally with a new technology, there's a lot of questions about, are you just identifying nonsense, right? And we were very concerned that we were identifying nonsense. And so we did a bunch of work to look at these in situ using single molecule RNA fish, which you bind a probe to primary RNA transcripts and then visualize it in situ. And we're able to confirm that we see a bunch of these cells that are single positive for SCGB3A2, and they appear to be in kind of these crazy formations. And so what you can see here is kind of what you'd expect from maybe a normal airway where it's got kind of this structure and there's sort of cells lining it all around the outside. And what we see with these sort of single positive SCGB3A2 cells in red is that they're kind of sitting in a lot of these weird kind of cystic regions that aren't sort of normal airway structures. And they appear to be both in these cystic regions as well as in some of the more fibrotic regions, which I don't have a very clear picture of in this exact example, but we were able to identify them in situ in these lungs. But more exciting than these cells were this novel profibrotic epithelial cell. And so we identified these really early as we were starting to generate this data. And these are these cells that we called KRT5 negative, KRT17 positive. And the reason we called them this, which again is a terrible name and very hard to say, is because these are 2 kind of canonical markers of the basal cells of the airway. And they were positive for one and negative for the other. But more importantly, they were actually expressing components of the ECM like COL1A1, as well as a number of other sort of really actually producing matrix. And similar to these SCGB3A2 cells, we were able to find these sort of in vitro, and we were able to see that they were kind of sitting adjacent to these regions of high fibrosis or these fibrotic foci. and that they were actually sort of truly collagen positive, as well as being positive for these other epithelial cell markers. So this work was really changed the way we and many people in the field were sort of thinking about the cellular composition of these diseased lungs. And there's a lot, we kind of came up with some theories about where these cells might be originating from, but there's still kind of a lot of debate about where they come from and where they're going, and are they an end state or a transitional state. And there's been a bunch of mouse work to sort of validate these cells. And they've now identified these cells in a number of other contexts, with individuals with pulmonary fibrosis. And the one thing I neglected to mention here is that these cells were found almost exclusively in individuals with pulmonary fibrosis and never in controls or individuals with non-fibrotic lung disease like COPD. And so since we published this paper, like I said, they found it in a bunch of other contexts, including in individuals who have lung scarring as a result of COVID infection. And so that tells us that they're kind of very rapidly occurring, and it's not a process of maybe this kind of longer chronic disease. So what was really interesting, just sort of as an aside, is as we were doing this work, we realized that there was another group that's sort of running in parallel who had found like almost exactly the same findings. We went to a conference, we were reading through abstracts, and this researcher, Naftali Kaminsky, had like described this exact same cell population, even using the same marker genes that we were using to describe it. So we actually sat down with Naftali at this conference and made a plan, and we ended up, instead of sort of competing with each other, working together, and we submitted both papers to bioRxiv on the same day. We submitted both papers to the same journal at the same time, and they were actually published back to back in the same issue of Science Advances. So it was kind of a fun story from a perspective that we ended up like not battling it out with each other. And it was really nice in the end to see that using a completely orthogonal data set that had been processed in a little bit of a different way, that these same cell populations were discovered there. And so ultimately, we kind of came up with this model of pulmonary fibrosis, particularly in the distal epithelium, where normally you have these sort of AT2 cells that serve as kind of a stem cell for damage, and as they're damaged, they repopulate the AT1 populations. And in these diseased lungs, you get this large accumulation of fibroblasts, and then instead of having this proper differentiation from AT1 to AT2 cells, you get kind of this aberrant differentiation pathway where they kind of create this profibrotic epithelial cell.
Quick question?
Yes?
So you're saying that the bottom layer, those things are arising from your AT2?
This bottom layer here? No. So I think these fibroblast process is probably somewhat different, but we think that the signaling that's occurring in this sort of distal epithelium as things are getting damaged is creating an environment that is conducive to this sort of profibrotic proliferation. But it's really, instead of these cells becoming the sort of type 1 cells like they should, they're kind of becoming this aberrant like profibrotic epithelial cell. All right, so like I said, this work is all published, and I'm happy to talk about it in depth if anybody has questions, but I think sort of more interesting is some of the, maybe not more interesting, I think this stuff's interesting, but more exciting for the sake of this talk is some of this new work that we're doing, looking at the genetic control of gene regulation using these single cell data sets. So this is work that's really led by Haney Natri, who's in my group, as well as Christina Izodi, who's actually just wrapped up her postdoc in Davis McCarthy's group at SVI in Melbourne. And so the goal of this study was to identify how genetic variation in individuals might be altering gene expression in these single cell data. And this was really a goal of this study from the beginning, but requires a lot of individuals to have the sort of statistical power necessary to carry out this type of work. And so like I told you, that cell atlasing study that I was just showing results from was 30 individuals. This study in the end, I think, had 116 individuals, so it just took us a long time to build up this data set. So to give you guys a little background on the idea of mapping genetic control of gene regulation, this is sort of studies that are trying to identify what are commonly referred to as EQTLs. So if you're sort of familiar with that literature, but the idea is, out in the world among people, there's a lot of genetic variation. You can almost think about it like a sort of random mutation experiment, that if you get a whole bunch of individuals at any given locus, there's going to be a bunch of different genotypes segregating. And the composition of individuals who are heterozygous or homozygous for a ref or an alternative allele is going to be different at any given locus that's not an LD. So individuals kind of have this really robust set of genetic variation that's out there. And so the idea here is that as the sort of natural genetic variation within the common genetic variation, greater than 5% minor allele frequency, that genetic variation will affect the level at which a gene is expressed. In some cases, if these single nucleotide polymorphisms sit in promoter regions or enhancer regions per se, and the idea here is that genetic change will maybe change the affinity of a transcription factor to bind to that site, and you could get either an increase or a decrease in gene expression. And so we like to map these interactions using this process, using sort of a regression-based framework. And so the idea is that any one of these loci, you aggregate all the individuals who are ref-ref, ref-alt, and then you look for a linear association between genotype and expression, and you do this for all genetic variants within some candidate window around your gene. In this case, we used, I think, 1 megabase, and then for every gene in the genome. And so we're interested in these because as people have carried out these genome-wide association studies, we've identified a lot of genetic variation that's associated with disease risk, but most of that variation sits in regions that are non-coding. And so the idea is that genetic variation must be acting to modulate expression rather than changing sort of the structure, the function of that protein. And so we try and take these EQTL maps and overlay them with these genome-wide association studies and understand how genetic risk is modulating disease risk. However, all these studies have been, yes?
That bottom right plot, is that for just one target gene, or is that for
So this is like represents a GWAS for a disease, right? So this, you could imagine in this case a GWAS for pulmonary fibrosis, where every dot here is a single SNP across the entire genome, and on the y-axis is its association with risk of that disease.
So you do that for the EQTLs, you do that for every gene in the genome?
Yes. Yep. Exactly. All right, so most of this work in the past has been done in bulk, mainly just because that's the tools that were available, and you have to get a lot of individuals, and it's kind of expensive. But we now have built this data set. We're able to do this at sort of cell type resolution, and the idea is that we believe you will have many cases where in cell type 1, a SNP does not have any effect on expression of a nearby gene. But in cell type 2, it will. And this could be because of different transcription factor usage. This could be because that gene is off in one cell type or on in another cell type. There's kind of a whole host of reasons why you might find this. But we really wanted to use this data to assess EQTLs at sort of cell type resolution. So like I said, we ended up generating data from 116 donors. We did single-cell RNA-seq and whole genome sequencing. Ultimately, we ended up with a data set of about 475,000 cells. You can see the demographics here sort of break down mostly European. The diagnoses are either individuals who are controls, individuals with pulmonary fibrosis, or we have a handful of individuals who have other types of fibrotic diseases that are not idiopathic pulmonary fibrosis, which is the worst form of the disease. And then you can see here on the right is this UMAP plot, and if you've ever looked at sort of any single-cell paper, you've seen a plot like this, but the idea is that every dot here represents a cell, and the distance between dots represents how sort of transcriptionally similar they are on kind of a fundamental level. And here we've color-coded them by their sort of cell type, and we've got the major sort of clusters of immune, epithelial, endothelial, and then mesenchymal there, kind of in the brown in the middle. All right, so to perform these EQTL analyses, we do something that's called a pseudo-bulk aggregation. And so the idea is for any given cell type, we take the average expression of all individuals, all cells from an individual within that cell type. So you can say we have T cells, and within T cells we have 50,000 observations of T cells, and we have 116 individuals. And so for individual 1, maybe 300 of those T cells came from them. We take the average of those 300 T cells, for the average expression of every gene for those 300 T cells, and that becomes their value. So in the end, you end up with this matrix where every column is an individual, and every row is a gene, and we have this same matrix for every cell type that we looked at. We did require individuals to have greater than 5 cells before we did a mean. Based on some of our simulation work, this seemed a place where we could get a reasonable enough estimate of averages that we'd have power to sort of do this work. And then here on the right, I'm showing the number of individuals who ultimately met this criteria, who had greater than 5 cells. And so you can see cell types that are more rare, like PDCs, tend to have far fewer individuals who meet this criteria than a cell type like monocyte-derived macrophages on the top left, who basically every individual in our study had enough cells to be included in that. So we perform this mean aggregation. We use a linear mixed model to identify these cis EQTLs, and then we use this approach called multivariate adaptive shrinkage to sort of estimate effects across cell types. And this was an approach developed by Matthew Stevens' group out of the University of Chicago, and they developed it for analysis of the GTEx data with the idea that by leveraging sort of structure across tissues that you could identify genetic effects that were shared across tissues versus sort of specific to an individual tissue.
Nick, can I ask a question?
Yeah.
So for isolating the cells when you had control versus other fibrotic tissue, did you see sort of some correlation or bias in cells that are buried deeper in the matrix for what you got out of control versus like denser fibrotic tissue?
Yeah. Yeah, so I'm going to repeat your question for people online, I assume. Yeah. All right. So Doug asked, do we see some difference in cells from the control individuals versus the individuals with pulmonary fibrosis, because there is a difference in matrix composition, and so some of those cells that are buried deep in the matrix in the fibrotic samples are harder to get out. And so the answer is it's a little bit confounded, right? Because we also have like cell type composition changes. So it's a little bit hard to tell what might be a bias of dissociation versus a bias from the disease. And so we tried to, when we did some of those proportional analyses that I talked about earlier, just looking at how cell type proportions change, we actually made the denominator the number of cells within a lineage. So if you had kind of a bias between epithelial and immune cells, that wasn't sort of throwing you off. It was only if you ended up having a bias kind of within epithelial cells that it would hurt you. But we're interested in looking at some of this in the spatial data where you don't have these same sort of biases, right? Okay, so as you might expect, if you think about statistical power, after we ran this sort of EQTL analysis, we were able to identify many thousands of genes that were associated with that had genetic control of the gene expression. But our power to detect these EQTLs was really driven by the sample size. So cell types where we had very low numbers of cells or few individuals who met that criteria of having greater than 5 cells per individual, we ended up having less statistical power to identify EQTLs. So not surprising, but nice to see that statistics work. We did find that using this analysis that most EQTLs in our data set were shared. And so we kind of have 3 classes. So here, on the left-hand side is a heat map where blue indicates high degree of sharing and sort of yellow indicates low degrees of sharing. And if you just focus on the bar on the top, which is kind of the high-level lineage area, where green is immune, blue is epithelial, sort of dark pink is endothelial, and light pink is mesenchymal, you can see that there's structure there in this data where things tend to be most often shared between between a lineage. And we kind of came up with these three different categories. So the first one is globally shared. So we found lots of EQTLs where if you're an EQTL in one tissue or one cell type, you're an EQTL in all cell types. We have these multi-states where it was less than global but greater than unique. So this again was ultimately like 64% of the EQTLs were at least in 2 cell types. And then ultimately we had a fairly small number, only 2%, that were really unique to an individual cell type. And we see that the characteristics of these EQTLs diverge a bit between classes, and there's some other ways that we've looked at this, but I think most notably EQTLs that are unique to a single cell type tend to be further away from the transcription start site, as well as have larger effect size overall within that cell type. And so I think the last point is probably a bit of sort of just statistical power issues. If things are really rare in the sort of adaptive shrinkage framework, you need a lot of evidence to say that you're really confident it's unique to a single cell type. So those tend to be enriched for these kind of large effect variants. But I think the distance to the TSS really sort of makes sense biologically. We think things that probably have really high cell type specificity are more likely to be things disrupting enhancers rather than a promoter region, because these tend to be the regulatory elements that are sort of have very differential usage between cell types or tissues. We can also look at, instead of just the sort of effect type, or instead of just the sort of degree of sharing, we can actually look at sort of individual, we can look at individual EQTLs. So here we've taken about 3,000 of these EQTLs, and here each column is an individual EQTL, and then red is its effect size, so higher effect size means that effect is kind of shared across, or more red means that it's shared across cell types. And then if you see blue, that means that in that cell type it sort of diverges from the common. So to sort of orient you here, you can see that in this sort of like thin middle strip here, you've got these effects where there's an effect in one direction in the epithelial cells, and you kind of see an opposite direction of effect in the immune cells. So it's significant in both cases, but with sort of inverse effect size, which is kind of interesting. For the most part, what we see is kind of these same sort of classes that we saw in the sharing plot before, where on the far left-hand side, you've kind of got a tranche of QTLs that are really pretty much shared across all cell types. Next over, you've got something that's mostly immune, but kind of at lower effect size. Then you've got a chunk of EQTLs that appear to be really specific to epithelial cells, another chunk that are fairly specific to immune cells, but with a larger effect size, and then this kind of smaller effect size epithelial cell here on the right. But we can see these sort of patterns of both sort of diverging and shared effects, and you can see that there's quite a lot of difference in some of these EQTLs across cell types. Yeah. So these are all the multi-state QTLs, not the specific. Yeah. All right, so ultimately we want to tie these back to these sort of genetic risk scores that we were talking about at the beginning. And so luckily there's a couple nice genome-wide association studies that have come out in the past couple of years for pulmonary fibrosis. One that is kind of an independent group that's done a big meta-analysis of a few other studies, and then another one that came out of the UK Biobank. And there's also a third that's a GWAS that's focused on East Asian population. So not quite as big of sample size and a different sort of genetic background. So what I'm showing here are on the left-hand side is a heat map of EQTLs that were significantly co-localized with GWAS risk variants. And we did this with the colloc package. And so for every cell type here, we're showing the number of EQTLs that were significantly co-localized with GWAS. And this is a pretty like rigorous approach to look at these overlaps that really not only looks at sort of significance, but tries to look at the sort of LD structure those p-values and really estimate if it's a true shared common variant in the EQTL and the GWAS. And so that you can see we do a pretty good job of co-localizing with sort of the meta-analysis, the UK Biobank, and we have quite a bit worse co-localization with the East Asian IPF GWAS. We also looked at 2 other non-fibrotic lung disease phenotypes, so child onset asthma as well as adult onset asthma. And if you look at these plots, the one thing that sort of popped out to us quickly is that a lot of the genetic risk in pulmonary fibrosis sort of segregates into these epithelial cells. And this is quite different than the genetic risk for childhood onset asthma, where much of that genetic risk kind of segregates into immune cells, which I think sort of logically makes sense given the sort of etiology of these 2 different diseases. If we take a less strenuous approach and just a less strict approach and just look at kind of the overlap of EQTLs that also sit in that are also significantly associated with in a GWAS, regardless of this sort of like shared correlation structure that's required by colloq, we can see that in general EQTLs are quite enriched among GWAS loci, and this is true across whether you look at any EQTLs, global, multi-state, or these sort of unique. And the top row is just a null set of SNPs, the same number as the number of EQTLs we ran through this analysis that are matched in their distance to the TSS. So you can see that most of these do not have a significant association with pulmonary fibrosis. So just to end on one kind of nice example here, this is one of the really strongly associated genetic risk factors for pulmonary fibrosis. It's near this gene called desmoplakin, DSP. On the left-hand side, you can see the Manhattan plots for the 3 different GWAS, the meta-analysis, UK Biobank, and East Asian. And then in the middle column, you can see that same SNP association with gene expression in these 3 cell types, AT2 cells, transitional AT2s, and AT1s. And then on the right are the sort of violin plots of these associations. And so for this gene in particular, it was only significantly associated with these 3 cell types. And these are 3 cell types that, again, are the cell types that make up that sort of distal alveolar epithelium. What's that?
Where is it relative to the gene?
Man, you asked me the important questions. I don't remember off the top of my head if it's, I think it's an intronic SNP, if I remember correctly. All right. So the last piece of my talk is going to be like the least well-developed, but is kind of fun, and we'll show some fun pictures here. And so this is some work that we've been doing recently in the lab using sort of spatial transcriptomics to analyze some of these same set of samples. So as Doug so astutely pointed out earlier, disaggregating this tissue to get cells out causes a lot of problems. There's cells that lyse, there's cells that are really robust to these processes, there's cells that might be sort of differentially embedded in matrix between disease conditions, and so this can lead to all sorts of sort of incorrect assumptions about what's going on when you're doing these sort of cell atlasing efforts. In contrast to that, these spatial transcriptomic approaches allow you to look at gene expression of sort of a variable number of genes depending on the technology sort of in situ. And so they're a really powerful sort of orthogonal tool to look at some of these same questions. So this is work that's being led by 2 people in my lab, Annika Vannon on the left and Ariana Williams on the right. So we've been using a new technology called the VisGen MERScope, but this falls into kind of a broad range of technologies that I'd call in situ spatial transcriptomics. And so there's a handful of companies out here. There's really good academic approaches to do this. But all of them work under the idea that you're imaging RNA molecules in high plexy within tissues. You're taking a thin slice of tissue and you're imaging them. So this is in contrast to some of the other orthogonal approaches that there's sort of an array-based approach where you take a slice of tissue, adhere it to a slide, anchor the RNA molecules from your tissue onto that slide with some sort of anchored probes, and then you sequence the sort of transcripts that are on that probe. So both of these approaches have sort of pluses and minuses. This is a targeted approach, so you're selecting somewhere between 100 to 1,000 genes, depending on the technology, but it's very, very high resolution, subcellular. The sort of array-based approaches, you're getting an unbiased view, so you're pulling down sort of all of the transcriptome that's capped or polyadenylated or whatever your sort of mRNA pull-down strategy is. But right now, the sort of commercially available approaches tend to have fairly low resolution. The spots are pretty big. There's quite a bit of distance between them, and some of that technology is improving. But again, they're sort of orthogonal approaches that both have their strengths and weaknesses. We've been using this approach partially because when we were ready to buy a piece of equipment, this was the piece of equipment that was commercially available to buy, so we bought it. So the way that this works, and this schematic comes from the company VizGen, but I think conceptually this is sort of true for all of the in situ approaches, although the chemistry may be a little different and they may get there a little in some different ways. But you take a thin slice of tissue, you adhere it to a slide, you anchor primary probes to your RNA species of interest. And so again, this can be 100, 300, 500, 1,000, depending on the technology. And then you attach secondary fluorescent probes onto that primary probe in a series of imaging steps where you attach secondary probes and you image and then you wash, and you attach secondary probes and you image and then you wash. And you do that for some n number of cycles, again, depending a little bit on the technology. And then you're building a barcode for each individual RNA molecule that you see on the tissue. And so at the end of your rounds of imaging, you know that this particular gene had a barcode of 11010, and I go to my code book and I say that gene was CD3E, or that gene was SFTPC, or that gene was desmoplakin. And you've got this sort of library of indices that match to your genes of interest. And then you get a pretty picture at the end. Well, more than that, but you do get pretty pictures. I've spent a really long time doing really, sort of visually unappealing genomics work where I've made lots of dot plots. And so the fact that I get to play around with microscopes just kind of makes me conceptually happier. I remember sitting in grad school and watching people in my program present videos on actin and myosin and feeling so jealous as I went up there and showed my QQ plot. So I'm just happy to be on the other side now. Okay, so again, why spatial transcriptomics? Why do we think this is important? So beyond the sort of caveats that I told you earlier about, for the pulmonary fibrosis, this is really a heterogeneous disease. So again, this is showing you a piece of lung from an individual with pulmonary fibrosis. And you can see you've got some areas over here that are incredibly highly affected. They're completely fibrotic. The alveoli are gone. You've got some areas down here where just chaos reigns, right? You've got these weird sort of cystic structures and maybe kind of airway-like structures, but they're all aberrant. And then you've got some regions up here and over here where maybe there's some remnant of proper-looking alveoli. So even when we're doing single-cell transcriptomics, we isolate all these cells, but we isolate them all equally from this sort of whole area. So if we see an AT2 cell, we don't know if that AT2 cell came from over here in this happy, healthy area, or if it came from over here where chaos reigns, right? So the idea of these platforms, and particularly for diseases like this, where you have this sort of very heterogeneous affect across the tissue, is we can really localize the things that we're looking at to these very specific regions of the tissue. So we've been working with an early release version of some products from this company that have allowed us to look at this with FFP. And so the way that we've approached this is we've made what's called a tissue microarray. So rather than running a single sample on a slide, we're taking 3 millimeter punches and arraying them and doing sort of 6 to 9 samples per slide. And so this is kind of what it looks like. The arrows I'm pointing to are actually those sections from that piece of tissue. And so we've been trying to kind of capture the diversity here within the tissue, where you can see the sort of circle on the left that ends up in the top right kind of has a mix of these more normal-looking structures and this kind of fibrotic piece, whereas this sort of piece in the middle really comes from that really heavily fibrotic and looks pretty sad. Okay, so here's what these data look like when you get them off the instrument. Every dot is an individual transcript, and they're color-coded by the gene for which they represent. So to give you guys a sense of what this looks like, we're going to kind of keep your eyes on this chunk right here, and we're gonna move in. So as we get closer, you can start to see sort of more resolution. As we get a little bit closer, you can see the blue color here represents nuclei, so you can start to get a sense. This is sort of 250 microns. And then as we sort of zoom into this sort of cell level, you can see that we're really getting sort of subcellular localization of these signals. And then these technologies, either sort of in a user-generated way or through the technologies, draw boundaries around each cell. Sometimes these boundaries are good, sometimes they're pretty sad. And then you partition the transcripts into the cell in which they're sort of bound. And so you end up with data that looks very similar to single-cell RNA-seq data in the end, because you have a cell and then you know all of the transcripts that came from that cell. So in this picture, this is showing the spatial data on the right, along with an adjacent H&E section on the left. And so you can see, while we're a couple microns away in the tissue, so there's some differences. Like here, the airways are sort of touching, whereas here, these two airways are a little bit spread apart. In general, we see that there's pretty good concordance between what we're getting out of the H&E and what we get from the spatial data. What's really fun is then we can analyze these using sort of our existing single-cell RNA-seq pipelines. And so this UMAP here I'm showing on the left is actually a composite from all of those samples on that TMA I showed you. So it's 8 samples total. And you can see that we get sort of not as granular of cell annotations as we maybe had in some of our single-cell data, but we still get pretty good representation. And like Doug mentioned earlier, one of the things we're really interested in is looking at how this cell census compares with the spatial platforms versus the single-cell platforms. And so we do see some like pretty strong differences. Mainly we see the alveolar epithelium, particularly the AT1 cells, are very fragile. And so they were really underrepresented in our single-cell data, and we see much better representation of those in the spatial data than what we saw in the single-cell data. But more importantly, we see as we sort of walk through the disease, going from sort of these control samples to regions that are less affected over to regions that are more affected, we see these cell composition changes that we expect. So namely, if you look at that sort of dark green, which is the alveolar epithelium, you can see that ratchets down every sort of rung you go over. The airway epithelium increases, the fibroblasts and immune cells increase, and so this is sort of does a good job of capturing the sort of cell census as we walk through. Okay, so we can also, this is cool that we can analyze it like single-cell data, but like we care about spatial things, right? We care about the sort of spatial localization. So again, I think these came off like a week and a half ago. So like the analysis is not super nuanced yet. But what was really interesting is just even doing the sort of thing that we all like to do, which is just scrolling through and plotting our data and looking at it. We really quickly found a couple of interesting things. So the first thing that we found that was really interesting, and the sort of little vignette that I'll show today, is this population of macrophages that appeared within the fibrotic foci in the disease, and were sort of appeared to be these like pro-fibrotic macrophages. And these have been described elsewhere in the literature, but haven't really been localized in the tissue. And so what I'm showing here on the left and right, green is a ECM gene called fibronectin, and then the pink is LYZ, which is a marker of macrophages. And so what you can see is that there's sort of these macrophages that are maybe kind of just distributed around sort of fairly heterogeneously. But then we've also got these dense clusters of macrophages here, kind of here, that are sitting in these really like dense fibrotic regions. and actually appear to be dual expressing both LYZ and fibronectin, so really kind of pro-fibrotic macrophages. And so what we could do is we could take this data that we had analyzed like single cell data and pull out this macrophage population. And we isolated this group of cells. We did a more in-depth subclustering and broke this original cluster into 3 sort of separate clusters based off of transcriptional similarity, again, in a totally unbiased way. So we just said, what are the clusters? And when we map those back to the tissue, we can actually see that those sort of subclustered cell states match back to this sort of information we were seeing in the spatial data, where this blue population of cells is these sort of pro-fibrotic macrophages, and you can see that the spatial distribution of those macrophages is much different than the other 2 populations in sort of green and this sort of salmon color here, where all of the blue macrophages tend to be really kind of clustered together, whereas the sort of green and salmon colored macrophages are sort of equally distributed. So all 3 of these cell types were in that population of macrophages, but we can see that they're actually mapping back to really spatially distinct regions. What I thought is really interesting, sort of as an aside, is there's almost kind of a line here where you go from like highly fibrotic to less affected. And these sort of like dense clusters of macrophages are sitting in the sort of start of these new fibrotic foci in the unaffected portion of the lung. So all the sort of really dense clusters of macrophages here in this plot are sitting in the regions that are less affected. Yeah?
So do you know if the macrophages in normal tissue look like this?
They do not. So if we plot these same cells into a normal sample, they all, first of all, we see very few of these blue cells in general, and then they plot much more like these other sort of salmon and green colors where they're kind of uniformly distributed across the tissue. And you do have in your lung resident macrophages that sit both sort of throughout your lung that are not monocyte sort of recruited macrophages. And so we do see those in these sort of alveolar structures. These? No, these are like classical sort of monocyte-derived macrophages. So the alveolar structures are not ciliated. They're made up of these AT1 and AT2 cells, whereas the insides of these airways are composed of a mix of ciliated and basal and secretory cells. So typically in these kind of bigger structures, which again, I realize you guys don't look at these plots obsessively like I do, so airways don't jump out to you, but these 2 regions here are both airways, and they tend to be lined by ciliated as well as other sort of airway cells. But as you get into the alveolum, the alveoli, the ciliated cells disappear. Yep?
Sorry, just one more on the blue cell type. Can you like take this data and then quantify that, like what percentage of all cells are this like blue macrophage?
Yeah, absolutely. So I mean, we haven't done those analyses, but yes, I mean, you can imagine putting them into a very similar type of plot to this, right? And breaking that macrophage population here in blue down into these components and looking at their distribution. All right, so finally, what we're trying to do with these data is really sort of think about molecular sort of histology, molecular pathology. And so we're in the process right now of a pathologist is reviewing all these slides. They're annotating all of there's a number of sort of distinct histologies that occur in individuals with pulmonary fibrosis. We're sort of annotating those across all of the TMAs that we've generated, and then trying to describe the sort of cellular composition and molecular sort of structure of those histologies. And so that, again, this work is all sort of very much like new and ongoing, but that's the sort of direction that we're moving in right now. All right, so I want to thank the people in my lab, and there's our most recent lab picture there on the bottom left. And then all of this work is in like very close collaboration with John Kropsky, who's an MD researcher at Vanderbilt University, and all of these lung studies are really me and him kind of hand in hand doing this work together. He's just like my favorite person in the world to work with, so he's great. And then we've also spun up a sort of series of collaborations with Davis McCarthy's lab in Melbourne, and he's a statistical geneticist who does a lot of sort of methods development for genetic and genomic approaches, and he's helped a lot with some of the new methods in the EQTL work, and then is also thinking about some of the spatial data as well. And for my funding and support, and happy to take questions. So, yeah, sorry. I haven't been repeating any of the questions. Yeah. Okay. Yeah. So the question was, with the subcellular approach, can we actually identify differences between transcripts that are in the cytoplasm versus the endoplasmic reticulum? And the answer is, I don't know for sure. We haven't tried to look at it in that depth. My interest in the subcellular has not actually been subcellular localization as much as my ability to really localize transcripts within a single cell, as approach to some of these, in contrast to some of these orthogonal sort of array-based approaches where you might have multiple cells overlapping a spot on your array. Yes, so that's a really good question. So the question is, how rare of a transcript could we detect using the spatial transcriptomics approach? The answer is, I don't know. We have some transcripts that appear to be fairly rare that we capture very well with the spatial transcriptomics. And then there's some transcripts that should be fairly common that we don't capture very well. And so I think much like some of the early days of single cell, we haven't really built an intuition yet on exactly what's going to work and what's not going to work. But for example, there's a gene MUC5AC, which should be like fairly high in these lung samples, and we see almost no MUC5AC expression in our spatial transcriptomics data. And then there's another handful of genes that we expected to be expressed pretty low that we actually get fairly good representation from. I think we could end up with kind of similar sparse data matrices like we had from the single cell data, where you might have to get a composite of a bunch of markers to do this level of annotation. And I think to some degree it'll depend on the size of your library in which you're able to query, that as we move more and more into these 500 and 1,000 gene panels, this data was all from a 140 gene panel, but as we move into these larger panels and you have more orthogonal markers for the same types of cells, I think we'll have a better ability to resolve some of those differences. We certainly see less ability to resolve even major cell type categories in our spatial data than we do in our single cell.
Can I ask one follow-up?
Yes.
Do you think that subcellular localization, if you get more of the heterogeneous nuclear RNA before it's processed, do you pick that up better than that which is in the cytoplasm? Or things that are co-translated as they're moving through secretory, is that less easy to pick up?
That's a good question. I really have not, like I said, I haven't tried to do any partitioning yet based off of where we're seeing it within the cell. So I just don't have a good, I don't have a good intuition yet for how some of those internal cellular processes might drive this. I do think, the way these probes are designed, that if you have unspliced introns that might be breaking up places where you're anticipating your probe combine, you could end up with under capturing some of the nuclear RNA compared to the cytoplasmic RNA. But again, what we really need to do is sort of denote nuclei versus cytoplasm and compare what we're seeing within and without. And I know some of my colleagues who are more interested in like neural research where there's even probably a bit more work just on how subcellular localization of RNA might impact function, are spending more time thinking about sort of subcellular localization.
I'm going to ask one question. I don't know if there are any more after this, but I want to tie parts 2 and 3 together.
Yep.
So some of those QTLs that are cell-specific, do you see any in the monocytes that might be linked to these prefibrotic monocytes that you're finding?
That's a good question. I don't have an answer. It's so early, right? Yes. But yes, we're certainly thinking about how we could tie this genetic data back into these findings. So I mean, the nice thing is that these are all on the same set of samples. So these are an adjacent piece of tissue to what we processed with the single cell. So for every sample that we did single cell on, we disaggregated some, ran single cell, cryopreserved some, flash froze some, and FFP fixed some. So these are, really well matched to that single cell data. Yeah.
Do you know if these macrophages are lung specific, and if so, could you envision immunotherapy?
So these look like they're monocyte derived macrophages from our first from our sort of first assessment. So I don't think these are like lung resident macrophages. I think these are probably being recruited. And so while immunotherapy is sort of definitely something people in the space are thinking about, I don't think it would have, it doesn't disclude an immunotherapeutic approach, but it's definitely not a lung, not necessarily a super specific lung feature. Yeah.
So this is way out of my wheelhouse.
Okay, let's hear it.
This is about the VisGen Meriscope technology.
Yeah.
So you're basically taking slices of a slice and you're compounding them onto each other to create a really complex image.
So it's one slice of tissue.
Right.
So it's you're taking a single piece of tissue and then adhering one set of primary probes and then a secondary set of probes that are fluorescent, and you're adhering them and washing them off and adhering them and washing them off. So it's still all on the same slice of tissue.
So there's no way you could look at a 3D structure eventually of it?
So,
Would
yeah.
that even be meaningful?
Yeah, you need, yes.
Come over to our lab.
Yeah, go see Doug. He's got this.
Would it be meaningful to be able to see the disease tissue structure, or is it more of a game of just detecting its presence in a sample?
No, definitely structure is really important. And Doug has a system that's an academic version of the same technology. So he built the scope, he built the controller.
The problem is it's like petabytes of data.
Yeah, it's a ton of data.
So you have to really want to do it.
Yeah. Yeah, and so if on this system, we've thought about it a lot. The problem is, if you don't have like a light sheet, if you don't have a system that can sort of assay a thick chunk of tissue at once, you're sort of taking adjacent sections, and especially with FFP, it's hard. Sometimes you miss a section, sometimes the section folds, so then you've got 10 microns missing, and so we've been thinking a lot about how much, and a human alveolus ends up being, because we were like, could we do a whole human alveolus, right? And I think we figured out it ended up being like 50 slices. I mean, it's something that you're like, it's a lot of petabytes of data and a lot of money yeah. No, I mean, it's really interesting, right? And so we've actually been thinking about trying to do it in mouse models where the mouse alveolum, their lungs are much smaller and all the structures are much smaller, and so we could get more sort of 3D representation.
All right, let's all thank Nick.
Yeah. Thanks, guys.